faithfully delivering value, exploring the world's best books. Hello, this is Sally's Book Talk. Today I'll be interpreting the book I Contain Multitudes with the subtitle The Microbes Within Us and a Grander View of Life. The famous American poet Walt Whitman has a line, "I am large, I contain multitudes." This isn't just a literary metaphor. From a biological perspective, it's also true. Inside each of us, there's a microbial army containing 39 trillion microbes. These invisible microbes are like a puppet master, influencing or even manipulating other organisms, including humans. Do you know how they shape animals' bodies? How do they create new species? How do they manipulate animals' sex lives? And how do they lure them into the abyss of suicide? This book, I Contain Multitudes, aims to reveal the awe-inspiring power of microbes to us. The author of this book, Ed Yong, is a science journalist for The Atlantic. His work often appears in National Geographic, The New Yorker, Wired, and other science media. This I Contain Multitudes is written by him. After its first publication, it immediately became one of the New York Times 100 notable books of 2016. The New York Times, Wall Street Journal, and Science Magazine jointly recommended this book. Mark Zuckerberg and Bill Gates are both avid readers of it. Bill Gates commented that this book is super interesting. Ed Yong continuously presents one astonishing microbiological insight after another, showcasing the pinnacle of science reporting. Now, I'll take you through 4 aspects to glimpse the wonders of the microbial world. First, how do microbes influence the growth and development of animals? Second, how do microbes help animals acquire survival skills? Third, how do microbes manipulate animal behavior? Fourth, how do humans utilize microbes? First, let's talk about how microbes influence animal growth and development. Our Earth has existed for 4.5 billion years. If we compress that into 1 year, humans appeared only in the last 30 minutes of the last day, while microbes started colonizing Earth as early as March. From March to October, microbes were the only life forms on Earth, including bacteria, viruses, fungi, and all other microscopic organisms invisible to the naked eye. By November, plants appeared on Earth. December, mammals emerged. Since animals evolved later, they inevitably interact and co-evolve with surrounding microbes, and even their growth and development are influenced by microbes. In ancient Greece, Aristotle observed that a navy fleet docked on the shore threw a large amount of pottery into the sea. Not long after, live oysters surprisingly appeared in the pottery, and oysters can't move by themselves. Therefore, Aristotle thought they must have grown from the mud in the pottery. Although this idea was extremely wrong, it remained popular for centuries. By the 1990s, the same confusion occurred again at Pearl Harbor. At this crucial naval base, as soon as warships dock for just a few days, their hulls become densely covered with a tube-shaped worm called Hydroides elegans, making the whole hull look like it's covered in a shaggy carpet. Where do they actually come from? It turns out that the larvae of oysters and Hydroides elegans are extremely small. They ride ocean currents and are dispersed across the ocean, with a single drop of seawater potentially containing hundreds of larvae. When they drift to a suitable spot for growth, they develop into adults and cease drifting. And the key to triggering their maturation is encountering a specific bacterium, such as for Hydroides elegans. If it encounters a microbe named Pseudoalteromonas luteviolacea, it quickly develops from larva to adult. Without this bacterial stimulation, it would never grow up. In the Hawaiian waters around Pearl Harbor, this bacterium is present, and warships provide an attachment surface where bacteria can multiply quickly, forming a biofilm that serves as a breeding ground for Hydroides elegans. Similarly for oysters, pottery easily accumulates bacteria, which stimulate oyster larvae to develop, causing the phenomenon of oysters appearing to grow spontaneously in pottery. Many similar marine animals exist, where their larvae can only grow and complete their life cycle after encountering specific bacteria. Odd. Examples include corals, sea urchins, shellfish, lobsters, sponges, sea anemones, seashells, and more. In theory, without microbes, these animals could face extinction. Microbes not only affect development during the juvenile stage of animals, but also continue to shape their bodies after they mature. Take this creature called a planarian, about 1 centimeter long, with microbes filling almost everything below its head. Planarians have incredible regenerative powers. Cut one in half, and both ends become fully functional organisms. The tail end even regrows a head and brain. Chop it into 10 pieces, and you'll end up with 10 planarians. This unbelievable regeneration relies entirely on bacteria inside the planarian. The bacteria lock in energy. Any fragment with enough bacteria can regenerate. But if there are too few bacteria, that fragment will die. That's why the head, which has no bacteria, can't regenerate. A tail can regrow a head, but a head alone can't regrow a tail. From oysters, Hydroides elegans, and planarians, we see microbes decisively shape lower animals' development. But do they affect higher organisms too? Absolutely. Scientists raised mice in sterile environments and found they survived, but were extremely fragile. These germ-free mice had underdeveloped intestines with leaky walls, sparse blood vessels, and cells lacking regenerative drive. But when scientists introduced Bacteroides that iota micron, a common gut bacterium, everything changed. Microbes activated multiple genes in the mice, enabling nutrient absorption, toxin breakdown, blood vessel formation, and mature cell development. Essentially, microbes taught the mice how to use their genes to build a healthy intestinal environment. Microbes similarly impact humans. Our immune systems can't properly form without bacterial help. For example, when infected, immune cells rush to the site, causing swelling, redness, and heat. This inflammation is a protective immune response. Without it, infections would persist unchecked. But if the immune response overreacts, these cells attack healthy tissue, friendly fire. So we must both activate and restrain immune cells.
Microbes help us accomplish both tasks.
For example, segmented filamentous bacteria activate inflammatory cells, while Clostridia activate anti-inflammatory cells. Our bodies rely on these bacteria to maintain balance and trigger proper responses. Scientists also discovered that a common gut bacterium called Bacteroides fragilis powerfully activates certain immune cells. Remarkably, just a single molecule on its surface can boost immune cell counts. This marks the first time we found that a single microbe, even a single microbial molecule, can enhance immunity. Having covered how microbes shape animal bodies, let's explore their more advanced functions beyond basic development. Now let's examine part 2, how microbes help animals acquire survival superpowers. People once thought the deep sea was lifeless. No sunlight, water temperatures reaching 400 degrees Celsius under extreme pressure. Yet at 2,400 meters below sea level, we discovered a rich ecosystem with biodiversity rivaling rainforests. Giant tube worms there stretched to human height, strangely lacking mouths, guts, or anuses. So how does it survive? It relies entirely on microbes. Near hydrothermal vents, where tube worms live, abundant sulfides exist. The worms filter seawater to collect these sulfides. Bacteria inside them produce enzymes that break down sulfides, extracting carbon to power their bodies, simultaneously expelling sulfur. This chemosynthesis means tube worms need neither digestive systems nor mouths. Bacteria provide all energy. This energy mechanism wasn't discovered until the 1980s. Previously, all life was thought to be sun-driven, photosynthesis creating energy for plants and animals. But in sunless depths, microbes created an entirely new energy system. While creating vibrant ecosystems in darkness is impressive, energy provision is just their basic skill. Now let's examine microbes' advanced capability. If you visit European apple orchards in autumn, you'll notice unusual green spots on the yellow-orange leaves. This is the result of bacteria called Wolbachia, which parasitizes moth larvae living in apple tree leaves. When autumn comes, Wolbachia releases hormones in the larvae that prevent leaves from yellowing and withering, creating small green patches to give larvae time to metamorphose into moths. Eliminate Wolbachia, and the leaves wither, killing the larvae inside.
Sounds astonishing, doesn't it?
The previous examples show microbes helping animals survive through their own abilities, but they have an even more advanced skill: gene swapping, allowing animals to rapidly gain new survival capabilities. This term might sound sci-fi, so let me explain slowly. In the animal world, our genes come only from parents. You can't get my genes, nor I yours. But in the microbial world, bacteria swap genes as easily as exchanging phone numbers. Sometimes they approach each other like mating partners to exchange genes. Other times, finding dead bacteria nearby, they simply pick up genes from them. For example, scientists mixed harmless bacteria with dead pathogenic bacteria, and the harmless ones suddenly turned infectious. The harmless bacteria absorbed genes from the pathogens and integrated them into their own genomes. If one bacterium innovatively evolves a new ability, its neighbors quickly gain the same skill. Microbes' gene swapping ability also lets animals rapidly acquire capabilities they never had. Take the global pest called two-spotted spider mites. They eat over 1,100 plant species, though plants typically deploy chemical defenses with various toxins. These mites are practically universal detox masters.
But their detox genes aren't native. They come from bacteria.
When targeting a new plant, resident bacteria in the mite find bacteria with detox genes, integrate them, and instantly unlock new detox abilities. This gene loading method isn't just for spider mites. Humans can do it too. For example, pure-blooded Europeans and North Americans often get constipated immediately after eating nori seaweed. Nori contains unique carbohydrates. Humans originally lacked enzymes to break these down, but Japanese people digest nori without issues. They acquired this ability from bacteria.
This bacterium, Sobelia galactinivorans, lives in oceans and specializes in decomposing nori.
Scientists theorize that centuries ago, Zobelia bacteria on nori were harvested by a Japanese fisherman who ate both seaweed and bacteria. The bacteria found themselves in a new environment surrounded by unfamiliar gut microbes. Using their signature skill, they swapped genes, transferring nori-digesting genes to human gut bacteriides.
The adults passed these microbes to children, giving Japanese people permanent nori-digesting ability.
Animal genes were long considered sacred, impenetrable territory, unchangeable after birth.
But in 2001, scientists identified 223 human genes shared with bacteria that can indeed transfer to animals.
Hearing this, your view of microbes has probably shifted. They shape animal bodies, aid development, and grant unique survival skills. They seem like friends, but the author clarifies, microbes are partners, not friends.
Beneath harmonious revelation lie conflict, selfishness, and betrayal.
Now let's explore part 3. How microbes manipulate animal behavior.
Microbes manipulate animals ruthlessly. Take Toxoplasma gondii, a microbe parasitizing cats, its ultimate host. When infecting other animals, it desperately schemes to return to cats.
Scientists injected Toxoplasma into lab mice.
Normal mice, whether they've ever seen a cat, instinctively avoid cat scent because the instinct for natural predators is deeply encoded in its genetic programming. But mice infected with Toxoplasma gondii actually burrowed into a box with cat urine and lingered there because the toxoplasma in the mouse wants to return to its final host, the cat. So it changes the mouse's behavior, which is truly chilling. Not just rats. If infected with toxoplasma, they also become attracted to the smell of cat urine. Scientists have also found that toxoplasma might even make humans lose their fear of death. One explanation is that toxoplasma's metabolites can bind to certain receptors in the brain, making people feel like they've taken painkillers. This produces a numbing effect, which might make people with self-harm or suicidal tendencies feel no pain when cutting their skin, instead feeling pleasure. However, cat lovers needn't worry too much. Our immune system protects us from toxoplasma's harm. Toxoplasma can harm fetal health. But pregnant women shouldn't overreact. It's excreted in cat feces, which is currently the only known way it can infect humans. Pregnant women just shouldn't clean the litter box themselves. Microbes can carry out similar schemes. There's another parasite in African waters that must enter flamingos' bodies to reproduce. So they first invade a type of shrimp, turning the transparent shrimp red. The red shrimp is very conspicuous in water and easily spotted and eaten by flamingos. Thus, the parasite smoothly enters the flamingo's stomach. Microbes can also manipulate their hosts' sex lives. Earlier, we mentioned a bacterium called Wolbachia, which interferes with wasps' androgen secretion. It converts all males in the hive to females, allowing them to reproduce asexually.
When scientists gave these wasps antibiotics to kill the bacteria, males surprisingly reappeared in the hive.
Similarly affected is a butterfly called the common crow butterfly, whose male embryos are always killed by Wolbachia, resulting in a female-to-male ratio exceeding 100 to 1. And why does this bacterium always kill males? Was it harmed by males? It turns out that Wolbachia can only be transmitted through the animal's eggs, not sperm.
So it tries every way to increase the female population, even forcing the host into asexual reproduction.
Even in sexual reproduction, this bacterium can manipulate fertilization. Only eggs infected with Wolbachia can be successfully fertilized. Otherwise, the chances plummet. No, this means only infected females can reproduce generation after generation, allowing Wolbachia to spread almost 100%.
But manipulating sex life isn't Wolbachia's most impressive trick. It can even create new species.
Scientists discovered two closely related parasitic wasps called Nasonia geralti and Nasonia longicornis. Although they look identical, they are two separate species. Because there's reproductive isolation between them, meaning they can't mate and produce offspring. The wasps carry different strains of Wolbachia, which compete when the two wasps mate, killing the hybrid offspring. When Wolbachia is removed with antibiotics, the reproductive isolation vanishes instantly, and hybrid offspring survive. Thus, scientists speculate that microbes can split them into two new species, meaning these wasps might have originally been one species. And due to Wolbachia, they became two species. One study estimates that at least 4 out of every 10 arthropod species are infected. Wolbachia-infected arthropods are the most diverse, abundant, and widespread group in the animal kingdom, accounting for 80% of all animals, including insects, spiders, scorpions, mites, and more. The author says that if Wolbachia infects 40% of them, it could almost be called the most successful bacterium in the world, or at least the king of land. So, through 3 parts, we've taken you to experience the wonders of the microbial world. Such a powerful microbial world, if harnessed properly, can produce unexpectedly magical effects. Now, let's see how humans utilize microbes for our benefit. The first application, a bit gross, is called fecal microbiota transplantation, which can treat stubborn intestinal diseases. There are over 500 types of microbes in our gut, closely linked to our health. Different microbes form a complex network of mutual feeding and support, where pulling one thread affects the whole, making some intestinal diseases very tricky. Ordinary medical methods can't cure them. For example, a bacterium called Clostridium difficile can cause severe diarrhea, pseudomembranous colitis, intestinal obstruction, and other illnesses. In 2008, a 61-year-old American woman infected with C. diff and agonizing diarrhea. Her gut microbial diversity was nearly destroyed, and her weight dropped to just 25 kilograms. Doctors exhausted all treatments with no success. Finally, a doctor proposed fecal microbiota transplantation, transferring healthy donor stool to the patient's intestines. This would introduce a complete set of healthy microbial communities into the patient's gut. The woman's husband donated stool. Doctors blended it and delivered the slurry via colonoscopy.
Miraculously, her diarrhea stopped within 24 hours.
Within a month, C. difficile completely vanished.
No recurrence, a stubborn disease cured rapidly and completely.
This case became the prototype for fecal transplantation. Post-transplant, microbes reset her gut environment like an organ transplant. Successful cases multiplied. By 2013, clinical trials showed a 94% cure rate for C. diff with this method. Such high cure rates with no severe side effects are unprecedented in medicine. Even better, the treatment material is free. This marks a major innovation in microbial therapeutics. A nonprofit even runs a stool bank. Screened donors provide material that's filtered, encapsulated, and frozen for hospitals. Microbes can treat gut diseases and combat infectious diseases. Now for the second application, using microbes to eliminate dengue fever. Dengue fever, spread by Aedes aegypti mosquitoes, causes fever, headache, rash, and severe joint or muscle pain. No vaccine exists, leaving prevention as the only control method. Insecticides have limited effect against these mosquitoes and rarely eradicate them. So scientists devised a counterintuitive strategy: release more mosquitoes to stop dengue transmission. But these are special mosquitoes. We've implanted bacteria in them.
It's Wolbachia, mentioned earlier. Scientists found a strain that makes mosquitoes resistant to dengue virus.
Meaning, if mosquitoes carry this Wolbachia, they become immune to dengue and can't transmit the virus. If we could transform the entire mosquito population to carry Wolbachia, we'd cut off dengue's transmission chain. But how to transform the whole population? Scientists release enough Wolbachia-carrying mosquitoes into the wild. As explained before, Wolbachia spreads through insect eggs, ensuring only infected offspring survive. When these bacteria-carrying mosquitoes join wild populations, they infect others. Their offspring all carry Wolbachia, making them dengue immune. Experiments confirm this actually works. In 2006, scientists released Wolbachia mosquitoes in 2 Australian neighborhoods, dozens every 4 houses, totaling 300,000. Within 4 months, dengue-resistant mosquitoes replaced 90% of the native population. This marked humanity's first success in altering wild insect populations through engineered symbiosis to prevent disease spread. These applications might sound like sci-fi tech. Our third application is something natural and evolved: breastfeeding. We know breast milk is perfect infant nutrition, but do you know why?
Nutritionally, breast milk is just fats and lactose, similar to formula.
Its uniqueness is 200 plus human milk oligosaccharides, HMOs. Human milk contains the most HMOs among mammals, 5 times the variety and hundreds times the quantity of cow's milk. Scientists originally thought this was breast milk's key nutrition for infants, but the truth was shocking. Babies don't digest HMOs at all. After drinking breast milk, HMOs pass straight through the stomach and small intestine to the large intestine without being absorbed. It turns out these HMOs aren't for feeding babies, they're specially designed to feed bacteria. A bacterium called Bifidobacterium infantis in babies' guts devours HMOs completely, while other bacteria barely touch them. Thus, breast milk has evolved specifically to nourish Bifidobacterium infantis, helping it dominate the gut microbiome. When Bifidobacterium infantis digests HMOs, it releases short-chain fatty acids that feed intestinal cells, seal gut lining gaps, and produce immune-regulating anti-inflammatory molecules. So, if given other lactose sources, Bifidobacterium infantis survives but can't function properly. That's why breastfeeding is strongly recommended when possible. Breastfeeding is a microbial management system evolved over millennia. After weaning, we must independently nourish our microbes. As diets diversify, we cultivate more gut bacteria species, so Bifidobacteria lose dominance.
Unnoticed by many, modern humans' gut microbial diversity is declining, vanishing quietly like endangered species.
The main reason? We're not feeding bacteria properly. Doctors constantly recommend whole grains, fruits, and fiber. Actually, like breast milk, these aren't for our absorption but for feeding bacteria. Dietary fiber is complex, requiring multiple microbes with different enzymes to break down. It feeds diverse bacteria species. Without fiber, hungry bacteria scavenge alternative food sources. For example, consuming your gut's mucus layer, which also impacts microbial diversity. Experiments show mice fed low-fiber diets for months experience drastic gut microbial diversity loss. When high-fiber diets resume, microbial diversity rebounds. But it can't fully recover. Some microbes forced out never return. Passed down generation after generation, more and more microbes leave. Our research shows that compared to farmers in Uganda, Malawi, and Venezuela, Westerners have shockingly low gut microbial diversity. Ugandan farmers consume 7 times more fiber than Westerners. While Westerners also eat vegetables, most are highly processed foods where fiber is destroyed, and lacking microbial diversity means facing health risks. The author says the list of diseases related to microbes is unbelievably long.
From periodontitis to heart disease, from atherosclerosis to Alzheimer's, it covers almost everything.
A website contributor wrote, "In fact, nothing has played a more crucial role in our health than microbes." That's the main content of this book. The genius American director Orson Welles once said, "We are born alone, live alone, and die alone." The author says, this isn't accurate. Even if we're physically alone, we're never lonely. Because countless microbes coexist with us, it's time to rewrite our understanding of ourselves and microbes with Whitman's poem, that is, I am large, I contain multitudes. All right, that concludes our interpretation of I contain multitudes. Listening notes are below the audio. See you tomorrow.